Hello all, I am making this video to help you with the assignment called Human Blood Cell Typing. Before I get started on this assignment about blood types, I just wanted to let you know that I'm going to give you this assignment for this week, and next week I'm going to give you another additional assignment, and it is going to be on blood types and the genetics of blood types. After that, I will be giving you no more assignments. That means the last couple of weeks in May, when we were in the classroom originally, we would be reviewing for the EOC, we would be cleaning mannequins, things of that nature. I'm going to give you the last couple of weeks in May to catch up. If there's anything that you need more time on because you're going through personal stuff at home, or because you're taking AP and concurrent classes and you're covered up in work, you will have additional time to work on these assignments so that you don't get behind. Or if you just didn't get finished with one of those original assignments, just go back and do those. That way we can all get caught up and there's no hard feelings. There will be no essentially late assignments as long as they're in before my grades are due. So, have them in probably the end of May and we'll be fine. So to get started on this assignment does not require any additional internet research. You should be able to get all your answers from these questions from the assignment itself. So here we have Model 1, red blood cells and antibodies. And this table has a lot of information that you're going to be using to answer your questions. And so to go along with this table, this first column here talks about someone with blood type A. There's A blood type, B blood type, A, B, and O. But we're going to focus on A first. So, if you have A blood, you have A blood cells. And A blood cells have antigens attached to them. And antigens are just a specific type of protein. These are A antigens. So this makes this an A blood cell. Now, if you go and compare this blood cell to a B blood cell, you can see the only difference is the type of antigen that is placed on the outer part of the cell. These have A antigens, these little like spikes, and these have B antigens that make it kind of look like a little flower. Focusing on the A blood type again, whenever you have A blood, you make antibodies. Antibodies are part of the immune system, and they attach to specific antigens to try to mark it as foreign and for the body to take over and try to get rid of this invader, this problem. So with anti-B antibodies that are made by someone with A blood type, you can see that they have these little half moon structures. And so since A blood cells have spikes and these have like this little round structure, these antibodies are not made to attach to these A antigens. The shapes do not match up. And that's good. We do not want to make antibodies that attach and attack our own cells. You can see the shape of these anti-B antibodies, though, do match up with B antigens. So, for example, if a B blood cell was present with an anti-B antigen, these shapes would connect together and cause like an attack to happen. So I'm going to show you a little bit more here. We're going to skip to row number 4. Row number 4 says cells in the presence of anti-B antibodies. So we have A cells and we have anti-B antibodies. And as mentioned before, those shapes do not match up. And so the A cells are going to be able to flow freely through the bloodstream without any problems. And these antibodies are also going to be able to flow freely throughout the body. This is what you want. You do not want them attaching and attacking. Now here you have A blood cells, and they are in the presence of anti-A antibodies. So if we go look at anti-A antibodies, they have these tree-like, pointy-like shapes. Their shapes at the end of this antibody is made to attach to the A antigens. So whenever you mix anti-A antibodies with A blood cells, their antibodies will attach to the antigens, and it will cause the blood cells to come together. And this is what we don't want to happen. This prevents the blood from flowing freely through the blood cells, and plus the antibodies are essentially marking them for attack. That's kind of the basics of this table and everything that can really give you. The first questions they're going to ask are really basic questions. They're going to be easy at first, so don't overthink them. And notice they're going to say, according to Model 1. So this is Model 1. You read the question, you go up, you look at the table. According to Model 1, you read the question, go up to the table, and you get your answer. Everything you need should be on this table. To give you an example of a question that sometimes students overthink that's pretty simple, it says on letter B, what shape is used in Model 1 to represent an A antigen? So here is an A antigen, and some students will sit here and think about what that is. I would call an A antigen a triangle. It's triangular shaped, and so that's the answer. Don't overthink it, it's quite easy. Questions 1 through 6 all ask for you to go back to the model, reference the information, and answer the question. So if you have not done that yet, which I assume you haven't as we just started, I want you to pause and go through and answer questions 1 through 6, and then I'm going to help you with question number 7. So come back here for question number 7 whenever you're ready. Let's see. Question number 7. Suppose a person needs a blood transfusion after an accident. Blood is received from the donor. Note that the antibodies in the donor blood are not significant. That's really important. You'll see that in a moment. Only the antibodies in the recipient's blood will be considered. Use the diagrams in Model 1 to complete the following illustration. So, using proper terms, describe what would happen when someone with type A blood would receive blood from someone with type B blood. Now, I am very visual, so I have these little images here that you don't have to do anything with at all if you don't want to, but if you need them, they're here for you. So we just learned that someone has type A blood. So I'm going to imagine if they have type A blood, and I'm going to put everything they have. They have type A blood, they have an A blood cell. So I'm going to copy that and put it down here for my visualization. And if they have A blood, do they make antibodies? Well, I'm going to reference my model in case I need to know. A blood cells make anti-B antibodies. So, since they are the person receiving the blood, they would for sure have their own blood type, and they would also have their own antibodies. So there is our person receiving the blood. And it says when someone with type A blood, there we go, would receive blood from someone with type B blood. Now this part is really important. Whenever you're receiving blood, you only receive their blood cells and not their antibodies. So you would receive the blood cell of someone with type B blood. And if you have type B blood, you make anti-A antibodies. But whenever you're receiving that blood, it's like you only get the B blood cells. They do not bring any antibodies from the donor and stick it into the recipient's blood. So this would be the scenario then. Now the question is this compatible? Does this work? And so you have A blood cells and B blood cells with their A antigens and B antigens, and you have an anti-B antibody. So will the anti-B antibody and its shape, will it attach to the anti-A antigens? And you can see these are round little half moon shapes. These are spikes. No, they will not attach because their shapes do not match. Anti-B antibodies, though, can and will attach to B antigens because they're round shaped, so their shapes fit, and anti-B antibodies go against the B antigens, kind of how their name works. So what would happen is something kind of like this. I don't know if I'm going to be able to make it work for what I need it to work for, but these antibodies would come in and they would attach to the blood cell, and they would have multiple B blood cells here together, causing like this clumping action to happen, which is what we don't want. So you do not have to do any of these visuals if you don't want, but they help me, so I just put them there. It says here, using proper terms to describe what would happen. So what would happen is anti-B antibodies in the recipient with A blood. I want to put that in common. Would attach to the B blood cells. This means that A blood should not receive B blood. They are incompatible. Incompatible. So yeah, that's what would happen. If you want to leave your diagrams up here, you can. I'm going to be reading the descriptions. So this is what really matters. Also, it may be easier for you if you just draw this on a random piece of paper, in your own time, and then use that information and put it into words, whether or not the blood types are incompatible or if they are compatible. Sometimes it can be confusing, so please do not hesitate from sending me an email if you have any questions. You'll see that the next few questions under question 7, B, C, and D, they're the same type of question. It's just using different types of blood type to see if A is compatible with O, or if O can receive from A, so on and so forth. And then there's just a few more questions left about blood type. So hopefully you guys will do okay with it. Once again, please do not hesitate to give me a shout out if you are struggling in any type of way, because I want to help you. I definitely want you to understand this material, as we will use some of this information in MI next year. I do think this is important, so that's why I assigned this assignment. I think that's it. I hope to hear from you guys soon with questions or needed help.